All right, so the first part of this question deals with bonds and interactions in proteins. And the three types that we're going to talk about are covalent, hydrogen, and van der Waals forces. And the covalent bonds form strong bonds, and the reason for that is because the two atoms share a pair of electrons, so they both keep that electron, and it makes them very stable. And this type of bond is found in the primary structure of protein. And then there's hydrogen bonds, which form weak bonds. And of course, those form when there are hydrogen molecules present. And those occur when the hydroxyl group exists in a larger molecule. And this is the secondary, tertiary, and quaternary structures of protein, which are the alpha helix and the beta-pleated sheets, and also the tertiary, which is a combination of those two. And then the next type of interaction is van der Waals forces, and these help hold together nonpolar amino acid side chains, or R groups, and amino acids make up proteins, which is why this is relevant. And the interactions between these are weak and only occur when atoms and molecules are very close together, and this creates unequal electron clouds. And this is found in the tertiary and quaternary structures of protein, which tertiary is again that combination of alpha helix and beta-pleated sheets, and quaternary is any three-dimensional structure. Okay, so the next part of this question has to do with muscle contractions. So in order for a muscle contraction to occur, the actin and myosin are present, which are filament proteins. They're really thin. And then the tropomyosin and troponin, which are proteins, block the binding of the myosin to actin. And then calcium ions alter the shape of troponin, which allows the myosin heads to attach to the actin filaments. And then once they're attached, the contraction is achieved by the two proteins sliding past each other. And the myosin heads use ATP to attach the actin filaments and detach as well. And all of this occurs just so you can curl your arm up into a muscle. All right, so next we're going to deal with the regulation of enzyme activity. And it's important to remember here that the structure of a protein affects its function. And protein structure can be altered in several different ways, some of those being salinity, temperature, and pH. And these can disrupt the hydrogen bonds, ionic bonds, and disulfide bridges, as well as any other sort of bonds that form between proteins. And it destroys functionality. For most of them, of course, there are exceptions. But if you remember enzymes, they're the proteins that facilitate chemical reactions, and they're highly specific. Like usually one enzyme will go with only one protein, or substrate, if you will. And the shape of the protein or enzyme changes to fit better to the substrate, which is called the induced fit model. So as you can see in this bottom picture here, the circle curls around the edges because it's trying to better fit to the substrate to have the best possible reaction. Alright, so now we're going to deal with abnormal hemoglobin and sickle cell anemia, which of course is a disease. And the genetic basis of the abnormal hemoglobin is due to a point mutation in DNA. A point mutation is a base substitution leading to a different amino acid in the hemoglobin, which will code for a different protein. And the protein that it codes for is valine instead of glutamate. And what that does is that changes the normal red blood cell shape to be sickled because of a lack of oxygen. And sickle cell anemia is found predominantly in regions where malaria is a common threat, so a lot of tropical environments, especially African nations. The altered hemoglobin leads to oxygen deprivation, which minimizes malarial infections. So therefore, those with the sickle cell condition are protected against malaria. Because this is such an advantage to them, the trait is passed through the heterozygotes to the next generation, even though actually having the sickle cell anemia trait is not good. As you can see here at the bottom, you have the individual who is homozygous recessive, which means that he would have sickle cell anemia, and he's crossed out because he's going to die of sickle cell anemia. But then he's got his brother, who is homozygous dominant, and he's going to die of malaria. So you can see that those heterozygous individuals are the ones that are going to have the best survival rate and will continue to reproduce and pass on the potential to have sickle cell anemia.